Hi, my name is Ray. I work as a research technician here at BAS Lab, Stanford University, Biochemistry Department. I specialize in the chemical mapping of RNA structures. This is the general process of internal library prep or sequencing. When we get our synthesized internal library design pool from a vendor, it comes with single-stranded DNA. So we have to put it through this process called emulsion PCR. Essentially, is a emulsion of the PCR reaction with minimal oil. So the PCR reaction is trapped into these tiny bubbles. So the purpose for that is to quarantine different designs so they can have their own double-stranded DNA generation. So after emulsion PCR, we get our double-stranded DNA. We gotta use this double-stranded DNA to do an in vitro transcription. Just like how our body makes RNA, this is just done in a test tube. So we're gonna use this double-stranded DNA as a template to generate the RNA. So we get our RNA molecules. We need to refold it using magnesium ions to restore or to show off this interesting structure of the RNAs. So how are we gonna capture this structural information? How are we gonna take a snapshot of this structural information of the RNA? We're going to use chemical probing. So there are different kinds of chemical reagent we're going to use. In the case of shape reagent, here the example showing is the 2A3 shape reagent. Just like other shape reagent, you attach to the 2'-hydroxyl part of the sugar ring. For the base, that's also not base pair, which means this locus is more flexible compared to the base pair ones. So after we get this modified RNAs, we're gonna use that as a template for reverse transcription to generate our cDNA. So the cDNA will have a mutation base corresponding to the modified base of the RNAs. So this is the way we're gonna transform this chemical modification of RNA into a mutated cDNA. So we need to do a page verification of the cDNA. Essentially what happens here is I'm gonna run through our cDNA sample through this acrylamide gel. Different sizes of molecules, they're running at a different pace through the gel matrix. For example, smaller ones will run faster to the bottom, larger one will get a little stuck in the top. So here we take a look and we're just gonna cut and extract the cDNA in the right size. So this process is done to make sure that we get the right size of the cDNA and also to get rid of the impurity or off product that's generated through the reverse transcription. So we have our cDNA, we're going to do the amplification PCR to bring back, first off, the number of the molecules, also make it into double-stranded DNA. Again, this double-stranded DNA has captured all the RNA structure coming from the chemical modification in a form of mutated base here. So we're gonna use this double-stranded DNA sequencing analysis. After we get the sequencing data back, we'll run our own analysis to generate RNA structural information.